Hello, everyone, and welcome to the Biophotonics Discovery Podcast. Whether you're a seasoned researcher, a student just starting out, or you just love a good story about how light is used to solve big problems, this podcast is for you. We're making the discoveries in biophotonics accessible one episode at a time. And now I want to welcome our moderator and first speaker, Vanderlei Bagnato. Professor Bagnato holds a PhD from MIT and dual undergraduate degrees in physics and material science. A full professor at the Department of São Paulo since 1993, he is also affiliated with the Texas A&M's Biomedical Engineering Department. His research spans laser cooling of atoms and biomedical optics, and he has authored over 800 papers with more than 25,000 citations, and he's mentored over 130 graduate students. He is a member of several prestigious academies, including, but not limited to, the U.S. National Academies of Sciences and Engineering, the Brazilian Academy of Sciences, and the Latin American Academy of Sciences. He will start with a talk entitled Photodynamic Therapy, From Cancer to Controlling Infections Resistant to Antibiotics, an important choice on the global health scenario. He will also serve as the moderator following his talk and introduce the subsequent speakers. So with that, Professor Bagnato, please take it away.
Okay, Darren, thank you very much. It is a pleasure to be here. We always know how important is the global health and possible solutions for the global health problems and challenges we have. And just to make sure, I will give you the first talk, and then I will be the moderator. So without any further delay, I would like to start my presentation. And I will be speaking to you about the photodynamic therapy from cancer to control infection, especially the ones resistant to antibiotics, and how important it is for global health. So when we speak about global health, we have to be a little more specific, right? Because we have all kinds of problems. It's not just a few ones. We have more problems that we can deal with. And then we needed to somehow think about different necessities, not only the ones that are superficial available, especially because we use optics, but there are many other ones that we can do it. So we needed to give priorities without exclusion of the problems in global health. When we're speaking about global health, economic reality is a very important ingredient. If we produce technologies that are forbidden for most of the communities that need it, we are failing. The technologies in global health, the way we are proposing using biophotonics in general, they must be easy to use and to maintain, because most of the communities that need the new technologies, they don't have all the apparatus or the infrastructure to require to maintain technology. So you have to produce technologies that are robust enough, not only to solve the problem, but to sustain for a long time in use. And finally, we needed to have always together with what we propose, a platform of instructions and assistance. We should know that the reality of instructions and to use a new technology is not the same everywhere. And the presence of things that make sure we can always assist who is using is very important. And I would like to say that we needed to give support, but we should not restrict local team to operate and to give support to, because that's a way to grow globally, not only providing solution, but involving more people that in the future will be able to provide solution. I know that most technology deals with diagnostic, but we need to know that treatment is as important as the diagnostic. When we speak about global health, there are many, as I said, it's a big variety of things. Nevertheless, there are some choices, and people that are worried about the problem of global health, normally they take a few of the problems as challenges. In my case, I'm not going to speak all the problems, but the ones that I'm focusing in is the microbiological control, which is a bombastic problem, very big problem. The cancer has always been a problem, and chronic diseases, especially the ones related to pain, either in child or in adults. And more recently, we've been challenging ourselves to see how can we deal with the problem of food security, especially the fungus that's present in food and that's causing a lot of diseases. What we use for that, of course, biophotonics. Biophotonics is something that it is unlimited the way we can use it. And photodynamic is one of the most important ingredients I use in my development. And we all know that photodynamic is relying photoreaction and production of radicals, free radicals that can destroy locally where they are produced. But we cannot neglect the use of UV, which is much before photodynamic, metabolic control, and of course, more recently, the use of sonochemistry in conjunction with photodynamic. If you can improve the reactor that light can produce, why not? And I'm going to show some examples that we have done very well in improving, combining sonodynamic with photodynamic in many cases. In terms of skin cancer, we treat all kinds of cancer, of course. Photodynamic is a useful technique if you have the right instrumentation and the right conditions to apply. But I've been focusing skin cancer because skin cancer, as I say, is not deadly but costly. Only in Brazil, where I live, we have 300 new cases of cancer, skin cancer, non-melanoma every year. In the United States, this is even overpassed. And everywhere in the world, you either have non-melanoma or melanoma skin cancer. And photodynamic helps a lot. This is the instrument we have been developed in Brazil, now is in many countries in Latin America, and we hope to go around the world because it is a fluorescence-assisted technology. So by looking at the fluorescence, you can guide in real time how you're doing with the treatment of the skin cancer. We use mostly topical photosensitizer in this case. We start with a success of 83% in a 5 years follow-up, but now we are reaching 95% in 5 years follow-up, which is very good because the best surgery you can have for skin cancer reaches about 97, which is our target. And we believe one of the problems is the drug delivery, so we've been doing well with the drug delivery. Now introducing the soluble microneedle. You can read about this on the paper that Physics World published and was news in the cover of the journal. And what we do is microneedles that stick into the skin cancer. They are about 1 millimeter, but they are micro in size, and they penetrate, and they dissolve. So we can overcome barriers of cream penetration and stimulating the cells to produce the PP9. And then we hope to reach about 97% on the treatment of skin cancer. And then we've been doing many other different devices for that, including devices that you can use at home after the first treatment in a hospital, followed by a medical doctor. And this device is being approved, and now in Brazil is approved by the social medicine. So we hope to have at least 2,000 centers doing free treatment of skin cancer, small lesions in Brazil in the coming year. Well, there is another problem, that's the cervical lesions, especially in Africa and Brazil, Latin America, and then in North America too. And photodynamic is especially good for all the levels of NAIC, the intraepithelial cervical lesions. And we've been treated level 1, that normally they don't do anything, they just observe. And by treating level 1 in a simple way, the procedure is very simple. It's a cream put it in the region, and the lady can put it itself, and then after a few hours, we do the treatment, and we being suppressed 70% of lesion of level 1 to progress to 2 or 3. We also we not treat only lesions on the cervix, on the uteral column, but also around it. And this is being a nice success. And devices is being approved, and now we are introduced this in Africa, hopefully together with the Global Health Program of NIH itself. And this is going very well. But we have also another challenge, which is the oral condyloma. We treat HPV in the same procedure of the NIC lesions, but now we have a big avalanche of lesions, oral lesions of HPV. And we develop a different way to do with PDT, with composed of a ultrasound scope that you remove the lesion and then you treat by PDT the floor of the lesion, and that we have been very well succeed much more than the conventional chemical treatment. Well, oral lesions is being part of many clinical trials, not only in Brazil, in different places. We're going to have a special talk about oral lesions, and this is being done in very good conditions, and even in big lesions, we've been doing quite well with interstitial, as you can see in one of the picture here, that you have a squamous cell carcinoma in the floor of the mouth, and we're doing well with PDT, with great success. Well, cancer is being one of the challenge, and this is being growing. And one of the reasons that did not grow before is because we need to put the conditions in a comfortable way. Equipment has to be connected with the medication in a way that always they are there. Otherwise, there is a mismatching of things. Now, related to the microorganism, we can speak about all those microorganisms, but I'm mostly concentrate in the respiratory tract, which is a big problem. We have now pneumonia, on basis on bacteria, virus, but now we have the fungus that is affecting the respiratory tract, and we are preparing to do that with big success. In our way to do that, we've been decontaminating mouth for dental procedures. Implant is not a small size procedure. Normally, there are surgery involved in doing implant, and one of the reasons of failure in dental implant, is contamination. And we've been doing this. This is a rinse of the mouth and a light that can eliminate most of the microorganisms, and you prevent that disease. But this is another good thing, which is the photodynamic for pharyngotonsillitis. As you know, the infection of the throat is one of that affects mostly the kids, and they have every year, and adults have every year too. And the way we proceed for that is, and one of the reasons is that is a very big consumption of antibiotics and making those infections resistant to antibiotics. So the way we did is we developed a syrup, a pastel, or a lollipop that kids, especially, is our focus here, but we are also treating adults. They use that's a hydrogel that is based on curcuminoids, right? A specific curcuminoid. And by doing that, we prepare the lesions, and after 5 minutes, 10 minutes waiting, the doctor can come and do the illumination. And in above 90% of the cases, we eliminate the lesion. And the rest of it, we prove that at least they are now back to be viable to antibiotics again. So doing the photodynamic, combining with antibiotics, may be one of the great things to do in the pharyngotonsillitis, because today everybody is experiencing more than one antibiotic to exterminate the infection. We are in a phase 3 clinical trial for this treatment, and this is going very well, and we want now to introduce that very fast in many different places, because it's a much easier way to eliminate the pharyngotonsillitis. But can we use the photodynamic to break down the resistance of bacteria to antibiotic? And the answer is yes. I don't have to elaborate in 12 minutes how big is the problem of resistance to antibiotic. And, we normally classify by the MIC, which is the concentration of antibiotic you need to inhibit the progression of the infection. And of course, when the bacteria became resistant, this number is moving to high values, sometimes 10,000 times more. And you cannot administrate 10,000 more antibiotic in a person. It's not possible. So then it is resistant to antibiotic. And we needed to move this MIC back. And we discover, and this is well documented in the literature, that by flashes of photodynamic with some of the photosensitizer that we use, we can break down the resistance. We don't kill the bacteria fully, but we can bring down the MIC below the break point. And then we've been doing that for many different antibiotics and for many different infections, and they all work quite well. So this is a big hope, that we use the photodynamic to break down the resistance of the bacteria, combining with the antibiotic, because after we break down the resistance, this infection will be again susceptible or again viable to be treated with antibiotic. We've been focused in pneumonia, which is a serious problem. Everybody in this audience has somebody that died of pneumonia in an intensive care unit because of the resistance to antibiotic. Of course, in mice, we can treat almost everything, as my friends say, and we have done that. The whole idea here is annihilate molecules that goes, overcome the frontiers or the barriers of the surfactant in the lungs, reach the infection, and by external to the body, illumination with infrared. Therefore, we use photosensitizer that are activated in the infrared. We can eliminate the infections, and we've been demonstrating this. We have demonstrated in pigs. Now we are doing the sheep, because sheep is a animal that naturally develop pneumonia exactly like humans. And then here in Texas, we prepare a special laboratory to treat pneumonia in sheep, either the non-resistant and the resistant. And all the devices being developed, and we are quite well advanced on this. And hopefully, we will be able to demonstrate that we can treat the pneumonia resistant to antibiotics using photodynamic in a very special way. But what I really believe is that, we don't need to throw away the antibiotics that are not respond, because with photodynamic combined, we make them a good instrument for fighting infection again. So my proposal is to really do it. This is what I'm doing in animals now, model. That's the combination of photodynamic to break down the resistance and the antibiotic to complete the work in eliminating the infections. I am in Brazil, we have Embrap, which is a translational financing, and I've been helping to put in the market many products with many companies, and we are looking for international partners to keep doing the development, and more than that, put that in the market. That's when our technology became really available. I have a lot of people that contribute, and I have a collaboration with many places. And I just want to thank you and say that I don't have time now, but we are fighting chronic diseases, and now we got a budget for the screening of the diabetes and treatment of diabetes, which I'm not going to elaborate in here. Okay, so the next speaker that we're going to have in this webinar is Jonathan Celli. Jonathan, it's a pleasure to have you here. Jonathan is a professor of physics and affiliate member of the Center for Personalized Cancer Therapy at the University of Massachusetts in Boston. His laboratory works on development and translation of technology for image-guided photodynamic therapy, as well as basic studies elucidating biological determination of tumor response of PDT. His lab has also made significant contributions to the use of bioengineered 3D cell culture models for assessment of PDT and other cancer therapeutics. He's currently a PI on the NIH NCI Found U01 project aimed at integrating optical diagnostic and PDT as part of the Screen and Treat platform for oral cancer in India. He also recently co-founded Photodynamic Solutions, Inc., which aims to advance clinical and commercial translation of PDT technology. Prior to join the faculty of the University of Massachusetts, Professor Celli was an instructor in dermatology at Harvard Medical School and at the Wellman Center for Photomedicine, where we received the NCI Pathway to Independence Award. So, Jonathan, you have the 12 minutes, and I will give a warning when we're getting close to the end, please.
Okay, great. Thank you. Thank you, Professor Bagnato, for the nice introduction, and to all the organizers for the chance to speak here. So, I'm going to talk about using photodynamic therapy to treat oral lesions in resource-limited settings. And 1 second, it's not advancing. Quick disclosure that I co-founder of a startup company related to some of this. In case I run out of time at the end, I just want to say some important acknowledgements now. This was very much a team effort, and everything I'm about to show really involved very important contributions from a lot of people at different places. So folks in my own group, Taba Hassan's group at the Mass General Hospital, Aligarh Muslim University in India is where a lot of the clinical work took place that I'm going to show. And we are now working with other clinical sites in India led by Dr. Kuriakose. And the work we're doing now has collaboration with Dr. Ron Liang at University of Arizona. And also want to thank NCI primarily for funding, especially through the Center for Global Health at NCI, which has been fantastic in supporting this. So as you just heard, cancer is a big global health problem. So cancer in low and middle income countries, LMICs. So I just like to emphasize these statistics that, cancer in LMICs is not just sort of a fringe concern or something. It's really most cancer globally. So 57% of new cancer patients and 65% of deaths annually. And these numbers are actually on an upward trend, actually, as you may have just seen on the bar graph in the previous talk. So early detection is important, but it's really only beneficial if treatment is available to meet the increased number of diagnoses which you get from screening campaigns. And it's really treatments that I'm going to be mostly talking about. So one example of a particularly dire cancer, global health, public health situation is oral cancer in India, where, there is just astronomical number of cases. So these are the most recent global CAN statistics, and it's really driven there by widespread chewing of tobacco-based mixtures. And the real challenge is to treat these early stage lesions that can either be precancerous or early stage cancer that look like the panel in the middle here before they progress on to become something that looks like the one on the right here. In India, there's just not sufficient availability of surgery and radiotherapy to most patients. There's not nearly enough linacs, and really surgery would be the primary option. But it's not accessible to many, many patients. So there's a real unmet need for a treatment that can be delivered at the point of care and that's feasible, preferably in rural health clinics and primary care settings. So in that context, we are looking at photodynamic therapy. So PDT involves light activation of a photosensitizing agent. There's two parts to this treatment. You have to have a light activated molecule that accumulates in the target tissue. You shine light on it, you activate some photochemistry, and actually a lot of photochemical reactions can occur, which I'm glossing over, but probably the generation of singlet oxygen is one of the most important that can lead to cell death in a manner which is very hard for cancer cells to become resistant to. So the basic idea is administer photosensitizer, or in our case, it's actually a photosensitizing precursor, aminolevulinic acid, ALA, which leads to accumulation, photoactive compound, and then you shine light at the target. So this is what it looks like in one of our own clinical images from India with the where the target lesion is in the oral cavity. You see the light coming in through the fiber optic there. Okay, so to do this all in a global health context, you want to make a solution that's ultimately going to be sustainable and can be affordable and be disseminated to the people who need it. So we explored using low cost light delivery with cheap LED sources and use image based treatment guidance and monitoring with a simple smartphone based system, which I'll talk a little more about, which has improved over the years. This was this first system from actually like 10 years ago now. But another important thing I just want to point out here is that when the same photophysics here, when you activate the photosensitizer, you also, in addition to sort of getting some quantum yield for generation of photochemical products like synthetic oxygen, you also get fluorescence emission, which is incredibly useful for treatment guidance and monitoring. So I'm going to talk about that a bit. So when we started working on this, we went through various design iterations of something that could be a cheap, low cost, portable, rugged light source that could be distributed. Went through various design iterations and came up with something that looks like this thing on the right here. I've had several variations of this over the years, but basically it's a high power fiber coupled LED with a touchscreen display. It operates on battery power. It's controllable through a smartphone app interface. And the light delivery into the oral cavity is enabled by little 3D printed pieces that attach onto the end of the fiber optic that look like this. So we package these into a little kit. And there's not enough time to explain how this works, but basically there's a piece that you bite down on, and then that attaches to a light applicator that couples to the fiber. And it's designed to be semi-customized by mixing and matching the different components to deliver light to different points in the oral cavity. So we did this first in the US, just in healthy volunteers, just to make sure that the system was ergonomic and usable, and then before shipping it to India. So the first clinical study that we participated in with this technology was at Aligarh Muslim University Hospital in India, in the radiotherapy department there, led by Dr. Siddiqui and Dr. Hussain. And we had important help from Stephen Bound and Colin Hopper from UCL, who are pioneers in PDT translation. So this was in just 30 patients with T1N0M0 stage 1 oral cancer with no nodal or involvement or metastasis, ALA photosensitization, treatment guided by smartphone imaging of the oral cavity to map the fluorescence, and also to assess the photobleaching after treatment, which acts as a reporter of how much PDT dose you did deliver. So the device delivered 100 joules per square centimeter, was calibrated to be about 50 milliwatts per square centimeter at the tissue surface. And it looked something like this. That's our hardware. Had gone from the bench to the bedside right there, and you can see light being delivered into the oral cavity. The clinical results were really encouraging. So we saw in most cases complete clearance of lesions in a single cycle of PDT that was durable and maintained at follow-up treatments. So 22 out of the 30 patients actually had complete tumor response. And as I mentioned, you can also look at the fluorescent signal to guide treatment. And one of the interesting things there is you see the extent of photobleaching measured at the time of treatment really correlated strongly with outcome, which is a useful thing we follow up on later. So that's not the end of the story. Yeah, I also wanted to mention the smartphone image guidance bit. So you can see the fluorescence from the lesion there, and then this dark spot here in the post-treatment image where the lesion has photobleached, and the patients that had really strong photobleaching like that actually correlated very strongly over here on the right in this bar graph, p less than 0.001 with outcome. So so as I mentioned, that's not the end. So one of the things that we started appreciating as we went through this is that screening is a big part of it. So our collaborator runs these screening camps where they go out through outreach and so go out into rural areas, and that's sort of how they would recruit patients. We started collaborating with Dr. Ron Liang at the University of Arizona, who was already doing oral cancer screening in India with a optical device. So he had a multi-channel autofluorescence imaging device that combines with machine learning to classify suspicious lesions. And we tried his device his oral imaging device, this handheld probe, in our photosensitized patients and found worked really nice for imaging the protoporphyrin 9 fluorescence. But furthermore, we thought that you could combine, this same dental camera form factor, you could combine the PDT light delivery. So you could have not only the, imaging for diagnostic purposes, but actually treatment therapeutic light delivery in the same device. Actually, this is the main focus of this Biophotonics discovery paper that was recently published. That's the sort of feature of this webinar, this special issue in Biophotonics Discovery. So you can read more about it there, but we're quite excited to deploy this in the clinic. So we're close to getting the approval to launch this at 4 sites in India, hopefully in early 2026. We call this the Screen, Image, and Treat optical system because it combines the screening capabilities of Dr. Liang's hardware with PDT light delivery and image guidance. And so far, so good with the preclinical data with this device, showing that we can sort of image and treat the same point. And so bottom line, the compact and low-cost light delivery and imaging systems that we've developed do enable use of PDT as a practical and effective treatment for oral lesions in challenging settings. And the new hardware, which I just showed at the end, integrates diagnostic screening with treatment planning and monitoring into the same handheld platform, and a multi-site clinical trial, which is funded by an NCI U01, is close to getting started. Going forward, I think a big thing is making it all sustainable. And so we've been working on tech commercialization through startup and partnerships. A recent award from the U.S. India Science and Technology Endowment Fund. So our startup is partnered with a company in India led by Dr. Pavan Gupta, also combining with machine learning for treatment guidance and automation of light delivery. And one last thing I just wanted to mention was that just because you develop a technology for global health doesn't mean it's useful at home as well. So here in the US, there's really no good non-surgical alternative for premalignant oral lesions that have very uncertain prognosis. And we believe PDT is actually a good fit for this and are working on developing this in the US as well.
Well, thank you very much, John. Well, very nice. You see, it's very important to have devices of easy use, right? And because guidance during procedure is as important as the diagnosis. A well-done procedure is, and photonic is helping so much for that, right? It's amplify the eye of the surgeon and the eye of the health professionals. Thank you very much, and we're going to have a question at the end. Now I would like to call our next speaker as Professor Jenna Mueller. Jenna is assistant professor in the Fitchell Department of Bioengineering at the University of Maryland, College Park, and in the Department of OB-GYN Reproductive Science at the University of Maryland School of Medicine. She's also a member of the program in oncology at the University of Maryland, Marlene and Stewart Greenbaugh Comprehensive Cancer Center. Her labs combine optical imaging, imaging processing, ablative therapies, and human-centered design to develop biomedical devices to solve challenges in global cancer. She received her bachelor degree in bioengineering with minor in global health technology from Rice University and received both the MS and PhD in biomedical engineer at Duke University for her work developing optical system and algorithms to improve the accuracy of cancer incision during surgery. Prior to joining the University of Maryland, Dr. Mueller was a postdoctoral associate at Duke University, where she worked with a multidisciplinary team to develop the POCT colposcope, a low-cost device to screen women for cervical dysplasia, which is now commercially available. Her work has been supported by NSF and by NSCI through different programs. Okay, I think Jenna is going to tell us about increasing access to laparoscopy surgery in low and middle income countries through human-centered optical device design. I think it's so nice that we are saying now, instead of low and middle income countries, we say low and income communities. Because as our previous speaker said, what we develop can be used not only for our countries, but for communities, even in developed countries, in well-financed countries, because they need. So, Jenna, please take the stage, and I will give a warning if necessary.
Great. Thank you, Dr. Bagnato, and thank you to the organizers for the opportunity to speak today. So today I'll be talking about our low-cost laparoscope that we've developed for LMICs, and I agree we should change this to communities instead of countries. And I'll talk about how we went through this iterative human-centered design process to develop our laparoscope. So our work is motivated by the lack of access to surgery in LMICs. This is a map that shows the proportion of the population without access to surgery, and upwards of 90% of the population lacks access to safe surgery in sub-Saharan Africa and in Southeast Asia. These areas of the world also have limited access to technology. This is a map of the world lit up at night, and as you can see, most of sub-Saharan Africa appears dark, even though there's huge populations of people here. And so if they lack access to our most fundamental technology, light at night, you can imagine they lack access to sophisticated biomedical technologies needed to treat a variety of diseases. Laparoscopy is widely used across surgical specialties that do surgery anywhere in the chest or abdomen. Laparoscopy is used for both diagnosis and treatment. And what is typically done is they insert this trocar port through the abdominal wall and then pump CO2 into the abdomen to create space to do surgery. And then through the center of the trocar port, they insert a laparoscope, which illuminates the abdominal cavity and provides a video to the surgeons to see what they're doing. And then they create a few more small incisions through which they insert other tools like graft spurs, scissors, and cautery tools to actually do the surgery itself. Laparoscopy is widely used because it has so many benefits over the traditional open approach where they have to create a large incision across the entire abdomen. Laparoscopy significantly reduces post-surgical mortality and morbidity. It reduces surgical site infections, decreases pain, and leads to minimal scarring for the patient. And also, it leads to a shorter length of stay, which is a financial benefit to patients. A lot of these procedures can be done through an outpatient approach, and patients can leave same day. But there are many shortcomings to the current technology, which is based on fiber optics and this laparoscopic tower. It was originally developed in the 1980s. And it's really expensive. It costs over $100,000 to outfit a single operating room with all of the laparoscopic equipment. It's fragile. It requires a lot of maintenance. A lot of times the light cords break over time and decrease light output. They're complex to assemble. They come in many different parts that then have to be each individually sterilized and reassembled. And the traditional laparoscopes can actually get so hot that if you touch the tip of the scope to the intestines, it can cause a burn. Or if you set it on the drapes, it can cause a fire in the operating room. So we wanted to learn more about laparoscopy in sub-Saharan Africa. And so we did a survey with surgeons at this conference in sub-Saharan Africa, and the surgeons are from a variety of different countries. And we asked them about things like the frequency of laparoscopy, and their interest in laparoscopy. And you can see that very few surgeons were able to use laparoscopy routinely in their practice, but a majority were interested in increasing its use because they know of the profound benefits that it has for their patients. When we dug in deeper about the perceived barriers to actually doing laparoscopic surgery, many of them were related to equipment. So there's poor access to equipment because of the initial cost and also the ongoing maintenance that's required. There's a lack of biomedical technicians to service the equipment, and they don't have access to sterilization a lot of times. Most of this equipment is sterilized either through autoclave or ETO gas sterilization, and that requires a significant amount of infrastructure in the hospital. And so instead, they actually prefer submersion sterilization in things like bleach or Cydex. And so 90% actually felt a low-cost laparoscope would serve an unmet need in their practice. And so this is what led us to develop our Keystone, which you can see pictured here. We can build it for about $1,000, and we've gotten rid of all of the fiber optics. This doesn't have any fiber optic components in it, and it doesn't require this laparoscopic tower either. So instead, we have placed consumer-grade electronics like a CMOS camera and a ring of high-power white LEDs in the tip of the probe. And then you just have electrical wires through the rest of the probe, and it connects to some circuit boards in the handle of the device. And it can be powered through USB. And so you can just plug it into any laptop, and we have a simple software interface, and that provides power to the device and the ability to view the video. And while we have this device now, that's not what we started out with. And we've actually learned a lot of lessons along the way. And we've used this approach called human-centered design to iteratively design this technology. So human-centered design is an approach to solving design problems that emphasizes end users' needs. And so you really need to have contact and observation with them if you want to actually design a solution that fits into their environment. And human-centered design traditionally has 3 phases: hear, create, and deliver. And so the idea is you hear what people say about their needs in your design, you create a new version, and you deliver it to the end user. And so in theory, this could be done once, but in practice, we go through this process many times. So I just want to illustrate a few iterations of the device to give you a sense of what we've learned. The first device we developed was at Duke in 2019, and we created this version that you see here with just a 3D printed handle. It was very simple. It just had this CMOS sensor here built into this stainless steel probe. And then actually in a separate device, we demonstrated LEDs. We just wanted to see if we could get enough illumination in the tip of the scope. And to test this, we used this setup on an optical bench where we have our device, and then we have this oblique illumination and then a target here. And this is set up for ISO guidelines. And we use different targets to quantify things like resolution, distortion, depth of fields. So how far back can you see before these lines blur together, and also color accuracy. And we published these results in 2021 with our initial Keystone. Here's just some representative results where we looked at a resolution target, and we also took an image with the standard of care system that was in the Duke hospital. And we looked at resolution, and we found that at short working distances, when we're up close to the target or to tissue, we do really well. We get comparable resolution to the standard of care system. But as we get further away, we don't do as well. And here you can actually see that we're not at the same limit of resolution as the standard of care. So here, lower limit of resolution is better, and we're not doing quite as well with the Keystone. And so around this time, I started my assistant professor position at the University of Maryland, and I ended up partnering with this Fischel Institute, which is an in-house engineering design firm that we have access to here to address some of these limitations. And so we worked with them to improve the resolution at longer working distances. We actually wanted to integrate the camera and LEDs, of course, as opposed to having them separate. We needed to decrease the diameter. We learned through a trip to Uganda, where we have a nice partnership there that I'll talk about in a minute. We needed to straighten the handle because surgeons like to hold this in different orientations. And we initially thought we just needed to waterproof the scope or the probe of the device so we could submerge it in Cydex. But we learned that we actually need to put the entire unit in Cydex, including the USB port. So we needed to waterproof the entire device in order to reprocess it in LMICs. So the new features, we worked on integrating this camera module with LEDs. We have 8 high-power white LEDs, a lens, a CMOS sensor, a sapphire glass window in this camera module that we developed alongside our industry partner, Misumi. We got this package down to a 5.5 outer diameter so it can fit in those trocar ports. We worked to change the lens system to have an optimal working distance of 3 to 7 centimeters. So here you can see we have improved resolution even at these longer working distances. And these are the range of working distances that are traditionally used in laparoscopic surgery. And we waterproof the entire design. So here you can see this is one of the trays that they use in the Ugandan hospital that we're collaborating with to sterilize equipment. And so this is just filled with Cydex. And we submerge the entire device. And now we actually are submerging the USB port as well. Here it was hanging out. But we have shown through repeated dunks that we still maintain a good working scope. But we learned that the LEDs weren't bright enough. And so we next created this Gen 3 device where we actually are pulsing the LEDs. So we're pulsing them at high frequency and they're really bright pulses, but we are syncing those pulses with the frame clock of the camera so that it always appears on to the end user, but we don't generate too much heat because in reality, the LEDs are actually off the majority of the time. And this is just an exploded view of the Gen 3 laparoscope. And we have all these O-rings that we helped waterproof the device. But the new feature that we incorporated in this Gen 3 is this custom PCB that allows us to do that pulsing of the LEDs. And so here we were able to increase the light intensity while keeping the temperature below this threshold where it won't cause burns. And we showed that even though we're not as bright as the standard of care system and will never be, we have a much more sensitive camera at the tip of the scope. And so we're able to actually illuminate the entire abdominal cavity sufficiently to get a good image. And so here you're looking at a pig in a study that we did where we imaged the intestines, the abdominal wall, the gallbladder, and the liver. And you can see comparable image quality with our device. We also are trying to figure out how to locally manufacture these devices in Uganda with our collaborators at Makerere University. And this is a shipping container makerspace that was created on campus. And we showed that we could actually assemble the devices and they would work in this makerspace. But during this assembly process, we learned that the soldering tools were not what we were used to working with at the University of Maryland. And because of that, they took so much longer to solder the camera into our boards. And so we figured out how to redesign the boards so that you could just plug the camera directly in. And that substantially reduced manufacturing time. And so now we're at the point where we're actually ready to do a clinical study at the Ugandan Cancer Institute. We just got all of our approvals in place, both at the national level and at the hospital itself. And so we're getting ready to start this clinical trial in early 2026, where surgeons are going to be using the keyscope to do laparoscopic biopsy, which is one of the simplest procedures that they can do in the abdomen. And we want to make sure that they can complete that laparoscopically without having to convert to an open approach, which is what they do now. And we'll track a lot of different outcomes like infection rates and collect usability data from surgeons as well. And so lastly, I would just like to acknowledge my amazing lab collaborators and funding primarily through the NCI and NSF.
Thank you very much. How important it is to make devices to help other procedures and everything, and the challenges you have to overcome during the way, things that you do not anticipate, you have to solve there. And now we have the last talk, the last contribution. And I would like you to welcome Nicholas Durr. He is an associate professor of biomedical engineering at Johns Hopkins University. At Hopkins, he directs the Computational Biophotonics Research Group. He's also a co-founder of Plan Optical, a startup that brought the Quick C auto refractor to the market. Okay, Nicholas, thank you very much for participating. And the title will be Improving Access to Eye Care with Computational Optics.
All right. Thank you very much, Professor Bagnato, and to the organizers for this webinar. It's really a pleasure to have the opportunity to share the work that I've done with and through Planoptica, as well as my research lab at Hopkins, to try to introduce tools that improve access to eye care. I have to start with some disclosures most pertinent to this presentation. I have a conflict of interest with Planoptica, and as you heard, I'm a co-founder and equity holder of this startup company. So to introduce the problem, I would like to talk about the need and opportunities in refractive eye care. So you may already appreciate the importance of prescription eyeglasses. It's widely considered one of the most cost-effective of all healthcare interventions. So you can buy a pair of prescription eyeglasses for just a few dollars in many parts of the world. And this can dramatically increase your access to education, your productivity, your quality of life. And so it has really far-reaching implications. But you may not appreciate that this remains a big problem worldwide. So there's right now over 1 billion people worldwide who have poor vision that could be corrected with a simple pair of prescription eyeglasses, and yet they don't have these eyeglasses. So we call these uncorrected refractive errors. It's a huge problem globally. And in many settings, this price point of prescription eyeglasses is actually affordable considering the benefits it provides. Instead, one of the major limitations is that there's not enough eye care providers to provide the specific prescription that each patient uniquely needs. So we have worked with Aravind Eye Hospital in southern India, one of the largest eye hospitals in the world, to develop technologies that increase throughput and the access to refractive eye care. In many settings, you can appreciate the demand by looking at ratios of eye care providers to the general population. So in high resource settings like the United States and Europe, there's approximately one eye care provider for every 6,000 people in these countries' populations. And in India, it's more like one per 100,000, and even worse in rural areas of these low and middle resource settings. So it's impossible for these eye care providers to provide the same level of care for these much larger demands with the existing technologies. And so I started working on this with some co-founders at Planoptica about a decade ago. And our original approach was we surveyed the existing ways to prescribe eyeglasses and measure what prescription you have, and learned that there's a technology called wavefront aberrometry that's very accurate, but unfortunately very expensive at the time and difficult to use and non-portable. So our mission became to try to translate this validated technology into a device that could be used in these low and middle resource settings to accurately provide eyeglasses just more efficiently. And the way that we did this was by using cheaper hardware, cheaper components, fewer components, and more advanced software, more advanced algorithms to convert these measurements into a accurate eyeglass prescription. The first few years of the project, we simplified the wavefront aberrometer to a minimal list of components. So this is already a decade old now, but this is the simple setup that we used. We started looking towards commercialization and called this the QuickSee. And it uses a low-cost camera sensor, a laser pointer, and a few simple optics in a see-through device. And then we record a video of spot measurements through a lenslet array on this camera that gives you a wavefront measurement that you can then convert into the ideal lens that you should place in front of your eye to correct your aberrations. So fast forwarding through several years of development to the startup company, this is what the first product looked like, called the QuickSee. And this quickly found use globally in hospitals, by optometrists, by NGOs in refractive eye care camps, and then also as screening settings through workplaces and schools and optical retailers. So we're very gratified to see this being used broadly now. There are millions of pairs of eyeglasses that have been prescribed with this device in many countries around the world. But if we take a step back and look at the leading causes of vision impairment globally, this turquoise bar represents the uncorrected refractive errors, and there's other causes of vision impairment. And really, to provide the highest standard of care for eye care, you really want to complete a comprehensive eye exam and not just measure the aberrations in your patient's eye, but also image the front and the back of the eye to look for cataracts, to look for retinopathies, macular degeneration, these other abnormalities and diseases that you should identify as an eye care provider so that you can refer these patients to a skilled practitioner for early treatment. And so the QuickSee right now, as I've described it, only provides refraction. And so my group at Hopkins has been looking into ways to simultaneously provide a refraction and image the eye. The inspiration for this technique came from Laura Waller's group, who introduced a diffuser cam. And similarly, Ashok Viraragavan at Rice University has been working on a flat cam approach, wherein you can image and reconstruct a 3D scene of an object using an untraditional optical element. Instead of a lens, you can use a diffuser or some kind of phase element in the optical chain. And so what we're doing here is, instead of using this lenslet array on the left, which is what is conventionally used for wavefront aberrometry, we introduced a diffuser, which produces as a point spread function this caustic pattern on the image sensor. And then for aberrometry, we can look at how this caustic pattern deforms in response to a non-ideal wavefront that's back reflected out of the eye. And if we register a caustic pattern with a flat wavefront, which represents good vision, compared to the eye that we're testing, you can convert this shift in the pattern to a wavefront slope, and then convert that into the Zernike polynomials to extract an eyeglass prescription from this measurement. And the advantage of this is that because this caustic pattern is so unique and it has so many complex features to it compared to a spot diagram, you can measure this displacement over very large movements across the sensor and get a large dynamic range for your autorefraction, meaning you can accurately measure the prescription for someone that has a high diopter of refractive error. And moving towards the imaging component of this, it's useful to compare it to conventional lens-based imaging. So with a lens-based imaging system, a point on the object is mapped to a blur on the detector, and this is your point spread function. And by contrast, diffuser imaging maps a point to an extended caustic pattern on the sensor. And importantly, this caustic pattern is linear and shift invariant. So as your point moves over your object, this pattern, this point spread function that you measure, is relatively shift invariant and linear. And it scales with the defocus aberration of your eye, or the magnification. And so what we do for this approach is we flood illuminate the fundus or the retina, and this pattern, all these point sources in the object are mapped to an extended an extended caustic pattern on the detector, and we measure a blur like this. And then we can essentially deconvolve this blur with characterized and measured point spread functions to reconstruct that flood illuminated object, that fundus or retina. And hopefully at the same time, get an eyeglass prescription out of this measurement in one device in one measurement. So for the study that I'm sharing here that was accepted into this special focus area for biophotonics discovery, we constructed one of these diffuser eye imaging systems where we measure, in this case, a model eye. We measure this blur on the left. We deconvolve it with a point spread function in the middle, and we can reconstruct the features of this model eye with a relatively short reconstruction and illumination time. And if we introduce aberrations or refractive error to this eye, we can reconstruct images with different point spread functions, taking at different defocus aberrations, and reconstruct a 3D shape of the eye, where we're essentially computationally refocusing this fundus image. So you can capture one image, and it doesn't matter how blurry it is from a defocus error. You can computationally refocus that image to make fundus imaging a little bit easier. And then we moved to the first in vivo human eye demonstration of this technique. So the way this approach works, we flood illuminate the fundus with 3 different colors. We calibrate our system. So we measure our point spread function at a variety of defocus errors with a pinhole and an LED. And then we deconvolve our measurements with these PSFs to get a focus stack. And then we can pick the fundus image that's the sharpest to get a refocused image of the fundus. And then that set point can also tell you about the refractive error of that eye. So hopefully with a little more work could be an accurate eyeglass prescription for that we extract from that refocusing technique. So here are the first in-human data of this computational ocular imaging approach. The conventional image is on the left. The diffuser measurement is right of that. And the reconstruction at different defocus errors is shown on the right. So this is, we're excited to show the first in-human fundus reconstruction with this approach. But you also see there's a lot of work left to be done to improve the quality of this computational image to look more like that conventional fundus image on the left. So in conclusion, I hope you can appreciate there's value from all of these presentations and trying to understand these clinical needs, these global health opportunities early on, and building something that's appropriate for the context that you're designing for. You saw from all the presentations, there's lots of parallel lines of work that must be done for impact to de-risk different aspects of the project. In my presentation, I've introduced computational techniques to try to improve these diagnostic tools, making things cheaper, faster, and working with a subset of components that you might normally use. And then we're here for global health, and we need to think about democratizing access to the technologies that we're introducing. I'll conclude here by thanking the team of researchers that have made this possible, especially the co-founders of Planoptica, Daryl Lim, Eduardo Laje, and Shivang Deve. The two researchers in my group, Marisa Marakis and Corey Simmerer, who led this Biophotonics discovery article, and then our many collaborators and funding sources. And thank you all for your time.
Thank you very much, Nick. It seems to be a wonderful process of coming with new technology, making the translation, and make sure that's been very useful for a variety of communities. Thank you very much. Okay, Darren, you want to say something?
I just want to say thank you to the speakers today for participating, and thank you to everyone who's attended. Just a reminder about the podcast and the special section, Biophotonics and Global Health, where there's papers, as you heard today, related to the topic here and others. Please don't forget to consider us when you're submitting. No APCs at least until mid-2026, and hope to see many of you at Photonics West. Thanks again, everyone. Bye-bye. If you enjoyed this episode, please subscribe, leave us a review, and share it with your colleagues. Your support helps us shine a light on the latest in biophotonics research. Stay curious, and we'll see you next time.